Hello, welcome to Rocky by Chem series. Please, if you've not subscribed to my channel yet, kindly support me by subscribing. Thank you very much. This video is about the Griffith Experiment in the year 1928. Frederick Griffith is a British bacteriologist who studied Streptococcus pneumonia, a bacteria that causes pneumonia in mice. In the ancient days, the scientists there used to think that bacteria are phased organisms and they are not capable of transforming or evolving. And from the studies carried out by Frederick Griffith, we got to understand that bacteria are capable of transforming. So, the finding from his studies has revolutionized the field of biology, and it has broadened our understanding of antibiotic resistance among bacteria. So, in his experiment, he used two strains of Streptococcus pneumonia. So, the first strain was smooth strain, which were virulent. There are two strains that he used for the experiment. So, Streptococcus pneumonia as a bacteria is responsible for causing the pneumonia disease, and there are two strains of the bacteria. The first strain is called smooth strain. We call them the smooth strains, and for these strains they have capsule around them, and because of the capsule around them they develop resistance against the host immune system. So the host immune system is not capable of protecting the body against this particular strain. So when they invade the system, they can cause the pneumonia disease. So the other strain that Griffith used in his experiment was the rough strain. As for the rough strain, it lacks capsule, and due to that, it is not capable of causing a disease. In other words, it is non-virulent. So in his experiment, he injected the first mouse with the virulent S strain, and after injecting it with the virulent S strain, he realized that the mouse dies. And then he moved on to the second mouse, and as for the second mouse, he injected it with the non-virulent R strain, and what he observed was that the non-virulent R strain couldn't kill the mouse, and then the mouse remained healthy. And then you move on to the third mouse, and as for the third mouse, he injected it with heat-killed S strain. He took a sample of the virulent S strain and then heated it to kill them. And after that, he injected it into the mouse, and then the mouse remained healthy. It didn't die. And then lastly, he got himself another mouse and then injected it with a mixture of the heat-killed S strain and then the non-virulent R strain. So after mixing them and then injecting it into the mouse, he realized that the mouse dies. So he then concluded from his studies that the non-virulent strain, as a non-virulent R strain, had been transformed into virulent strain by an unknown heritable substance from the dead virulent strain, and which became known as the transforming principle. It was a strange molecule for him. He wasn't sure of what that molecule actually is. He wasn't sure of whether it is protein, carbohydrate, or maybe DNA. So he just termed them as transforming principles. And his experiments opened ways for other researchers, including Avery, Macklin, and then McCarthy, for them to confirm what actually was the transforming principle. Kindly stay tuned for my next video about the Avery, Macklin, and the McCarthy experiment. Thank you very much. Please don't forget to subscribe.